Last month, I found myself at a quiet tech gathering in Palo Alto, sitting across from a man who looked like he was in his early 30s, lean, sharp-eyed, and just full of restless energy. We started talking about the future, and he casually mentioned his daughter's wedding in 1998. He wasn't aging gracefully. He looked like he'd been physically frozen in time. When I asked how he managed it, he didn't talk about exercise or clean living. He tapped his temple and said, "I just stopped running on legacy code." He spoke about the human body with this strange, detached fascination, as if he were reviewing a piece of poorly written software. To him, 2026 wasn't just another year. It was the great reset, the moment we finally moved from patching the rust to rewriting the source code of the flesh. He was practically vibrating with excitement as he described senolytics and epigenetic rewinding. But then, I brought up the FDA's recent push for more long-term clinical trials before these therapies hit the mass market. The change in him was instantaneous. The excitement vanished, replaced by a cold, simmering fury. He didn't call the regulators conscious. He called them agents of statistical genocide. He looked me dead in the eye and said that every hour a bureaucrat spends reviewing data is an hour where thousands of people are consigned to the grave by nothing more than paperwork. To him, the precautionary principle wasn't about safety. It was a slow-motion mass murder disguised as red tape. That jarring transition from the euphoria of a body reborn to a visceral hatred for the institutions meant to protect us left me stunned. It wasn't just a difference of opinion. It was a total rejection of the biological status quo. It made me realize that for people like him, we aren't just approaching a medical breakthrough. We're entering a war between those who accept the system crash of death and those who are ready to debug the human condition once and for all. Welcome to the deep dive. That tension, that almost utopian possibility of radical rejuvenation technology crashing headfirst into the very real, very slow bureaucracy designed to ensure public safety. That is our mission today.
It's a huge conflict, and we're going to unpack it by looking at the science, the regulatory hurdles, and, well, the pretty radical ways people are trying to get around them.
What's so compelling here is that the science, the foundation for all that euphoria you just heard about, is completely sound.
Oh, absolutely. The perception of aging has gone through this profound scientific and philosophical shift. It's moving away from being this abstract, inevitable thing.
And into the realm of a practical engineering problem.
That's exactly, something you can debug.
So let's unpack that, this fundamental shift. We're not just talking about aging as, the passage of time anymore.
Not at all.
We're talking about it as a system failure,
Yeah.
a debugging problem. So what is this geroscience paradigm exactly? What did it really redefine?
Well, the geroscience hypothesis is basically the consensus view now. It says aging isn't fate, it's not a calendar issue. Researchers define it as a systematic process.
A process of what?
Of deterioration, of our structure, our function, all the way down at the cellular and molecular level. You can think of it as accumulating entropy, really. And this deterioration, it shows up in a few key hallmark characteristics.
And what are the most critical hallmarks of that system failure? What should we be focused on?
The two that really dominate the conversation are genomic instability and accumulating damage to our DNA.
Okay.
Our cells are just constantly being bombarded, replication errors, environmental toxins, metabolic byproducts. And while we have these amazing repair mechanisms, they get slower over time.
Less efficient.
Right, less efficient. And the result is this continuous accumulation of damaged DNA and epigenetic alterations that fundamentally mess with how our cells are supposed to work.
So if the blueprint for a crucial protein gets corrupted,
the system starts manufacturing broken parts. It's that simple.
which leads to the big realization.
Yeah.
Aging isn't the effect of disease. It is the single largest cause of almost everything that eventually kills us.
Absolutely. That's the paradigm shift. Recognizing that aging is the root cause, the #1 risk factor for pretty much all the chronic diseases we associate with late life.
We're talking type 2 diabetes, cancer, heart disease.
Alzheimer's, generalized frailty, you name it. I mean, if you look at the economics, a staggering percentage of global healthcare spending is just dedicated to managing these age-related conditions. We've been fighting the symptoms.
The individual diseases as they crop up.
Right.
And that's the definition of the whack-a-mole approach, isn't it?
Yeah.
We've managed to keep people alive longer with statins for heart disease or certain cancer treatments, but we only delay the inevitable system crash.
Precisely. And that approach, it yields rapidly diminishing returns. You successfully treat one age-related disease, great, but then the person just succumbs to another one.
often something neurodegenerative like Alzheimer's or Parkinson's just a few years later.
It's an endless, incredibly expensive cycle of just managing symptoms.
And this is why the conversation has pivoted so forcefully from just extending lifespan to this idea of the health span imperative.
The focus is entirely on health span. I mean, that period of life where you're actually living with vigorous health, free of debilitating chronic disease.
That just makes more sense.
From a therapeutic standpoint, it's a game changer. Targeting the core biological mechanisms of aging gives you the ability to treat multiple age-related diseases at the same time.
So if you can slow down that underlying deterioration,
you significantly reduce the incidence of cancer, cardiovascular events, and neurodegeneration all at once. It's just a far more elegant and, frankly, effective approach than the old model.
So, okay, if the science is this compelling, if it's telling us aging is a treatable pathological condition, then we crash right into that regulatory wall.
Big one.
The fight over classifying aging itself.
Fair.
Why is getting the FDA to classify aging as a disease so existentially important to this entire field?
It's all about infrastructure and investment, plain and simple. Right now, the FDA's drug approval process is rigorously focused on single, clearly defined diseases. So if a company is developing a potential longevity drug, what we call a geroprotector, they're forced to seek approval for something specific, like reducing frailty in the elderly or preventing one specific type of cancer.
But the whole point of a geroprotector is its multi-disease benefit. So getting approval for just one of those things could take 5-7 years, cost hundreds of millions of dollars.
And then they have to do it all over again, sequentially, for the second, third, and fourth diseases the drug might prevent.
That sounds impossible.
That immense time delay and the staggering cost, that is the fundamental barrier to investment. Drug companies are just, reluctant to put up the billions required for trials because the return on investment is so slow and so fragmented.
But if aging were classified as a disease, the whole regulatory pathway would be transformed.
Completely.
What would that actually look like?
The market for these regenerative therapies would just explode. It would instantly expand to nearly the entire adult population. I mean, every person over 40, theoretically, could be a candidate for preventative therapy.
Which provides a clear, consolidated route to market.
And justifies the enormous R&D costs. It just changes the entire financial calculus. You go from a niche market to potentially the largest drug market in human history.
Now, the standard skeptical argument you always hear is that aging is natural. It happens to everyone, so how can it be a disease? It reminds me of past debates over things like, say, female pattern baldness or menopause.
And that's just a static, you might even say anachronistic, view of how we define disease. History shows us that what we consider normal changes all the time, based on our scientific understanding and what we can actually do about it.
You mentioned menopause. That's a great example.
It's a perfectly natural biological process, yet we approve hormone replacement therapy to treat its symptoms and effects. The FDA has already crossed this semantic line many, many times.
And we have even stronger precedents in medicine itself. What are some conditions that we used to just write off as normal aging, but now classify as pathological?
Oh, the list is pretty instructive. Conditions like osteoporosis, the degradation of bone density, or sarcopenia, which is the progressive loss of muscle mass and strength.
Both were just seen as, well, you're getting old.
Exactly. Just inevitable parts of growing old. Now, they're recognized as pathological processes with specific diagnostic criteria and, treatments.
And Alzheimer's is the big one.
For decades, Alzheimer's was just treated as severe senility, the inevitable cognitive decline of old age. We now recognize it as a distinct pathological neurological process. The goal here isn't just about changing a word, it's about defining new measurable biomarkers and, crucially, unlocking the potential for insurance companies to cover these massive preventative treatments.
So we're not just changing a label, we're unlocking the entire financial and scientific infrastructure needed to turn the promise of geroscience into a practical medical reality for everyone.
That's what the war is really about.
That brings us to the actual engineering solutions that are generating all this excitement. If aging is poorly written source code, we're now moving beyond just patching the system to actively rewriting the fundamental operating instructions. And the most compelling work seems to revolve around epigenetic reprogramming.
That's the real cutting edge. If you think of the body as having hardware, your DNA, and software, the epigenome.
The epigenome is the instruction manual.
It's the instruction manual. It tells the DNA when and how to express genes. Turn on the kidney cell genes, turn off the liver cell genes. Aging fundamentally leads to a kind of epigenetic noise.
The instructions get garbled.
They get garbled, and you end up with cells that are operating suboptimally. That's the legacy code your friend was talking about. So reversing biological age really means restoring the clarity of that original instruction manual.
And a key component in maintaining that clarity is the coenzyme NAD plus heredity. We hear this name constantly in longevity circles. What's its function, and why does it drop off with age?
Nicotinamide adenine dinucleotide, or NAD plus A, is absolutely fundamental. It's a vital coenzyme in every single living cell. Its main job is to fuel these reduction-oxidation reactions carrying electrons back and forth.
Which is essential for energy production.
In the mitochondria, yes. But crucially, it's also a vital substrate for enzymes involved in DNA repair, especially the PARP enzymes, and for a family of proteins called sirtuins.
So NAD plus is involved in both energy and repair. The dramatic drop we see with age, I've seen figures like 50% between youth and middle age. That must have massive systemic consequences.
It really does. As NAD plus levels fall, the ability of those PARP enzymes to fix damaged DNA is compromised. And the sirtuins, which are often called the guardians of the genome, they lose their ability to regulate cellular stress response and metabolism effectively.
So the cell essentially runs out of fuel for its own maintenance crew.
That's a great way to put it. This depletion is a core mechanism of aging, and manipulating NAD plus production, usually through precursors like NR or NMN, has been shown in animal models to prolong both health span and lifespan.
And that connects directly back to sirtuin activators, which have been a bit controversial in human trials.
Yes, sirtuin activators interact directly with that NAD plus enzymatic system. The most famous example, of course, is resveratrol.
From red wine.
From red wine, right. It gained initial fame because in early studies, particularly with yeast and mice, it showed remarkable longevity benefits. It even protected mice against the bad metabolic effects of a high-fat diet.
But the human results haven't been quite as dramatic or conclusive.
And that's largely a function of bioavailability. The doses you'd need to see the same effects in humans are often pharmacologically impossible to achieve because resveratrol is just poorly absorbed and rapidly metabolized.
So the underlying mechanism is still promising, but resveratrol itself wasn't the magic bullet.
Exactly. The pathway activating sirtuins is still a huge area of research, but the focus has shifted to finding more potent bioavailable analogs.
Okay, so if that's rewriting the software, let's talk about clearing out the junk that's already accumulated. That's the goal of senolytics. What is cellular senescence, and why are these cells causing so much trouble?
Cellular senescence is a critical part of aging. It's often called the zombie cell phenomenon.
Right.
Our cells, especially stem cells, are designed to divide a finite number of times. This limit is dictated by the shortening of telomeres, the little protective caps on our chromosomes. When they get too short, the cell enters a state of deep, irreversible arrest. That's senescence.
And historically, we thought that was a good thing, a defense against cancer.
That was the initial thought, yeah. Better a dormant cell than a damaged replicating cancer cell. But these senescent cells, they don't just sit there quietly. They're metabolically active and highly destructive.
How so?
They secrete this potent cocktail of pro-inflammatory molecules, growth factors, and enzymes. It's known as the senescence-associated secretory phenotype, or SASP.
And that SASP causes what we now call inflammaging.
Exactly. This chronic, low-grade systemic inflammation inflammaging is what promotes nearly all age-related conditions, from atherosclerosis to frailty and neurodegeneration. Senolytics are drugs designed to selectively kill these senescent cells while leaving healthy cells alone.
And this is where the animal data becomes truly staggering. It's why there's such an urgent push for human trials.
The results are astounding. Researchers at places like the Mayo Clinic used genetically modified mice where senescent cells had a kind of molecular fuse.
A self-destruct switch.
A self-destruct switch. When they administered a specific drug, it triggered those cells to die off. And in subsequent studies, they found that even a single treatment late in life resulted in a 20 to 25% extension of lifespan.
Wow.
And it came with measurable improvements in kidney function, fat mass, overall fitness. The possibility of essentially turning back the clock with one targeted treatment is why senolytics are arguably the hottest area in geroscience right now.
Let's pivot to another major regulatory pathway that offers a shortcut. The mTOR pathway. This is all about nutrient sensing and how our cells manage energy.
Right. The mTOR pathway mechanistic target of rapamycin is the cell's central metabolic regulator. It's the ultimate nutrient sensor.
So when nutrients are abundant,
mTOR signals for cellular growth, protein synthesis, proliferation. But when nutrients are scarce, the pathway is inhibited, which triggers the cell to enter a state of maintenance, recycling, and stress resistance.
Survival mode.
A survival mode that significantly extends cell longevity. This is the foundational observation that led to the discovery of the longevity benefits of caloric restriction.
So we know that severe caloric restriction works, but it's an extreme lifestyle. Rapamycin offers a chemical key to simulate that survival mode without having to starve.
Precisely. Rapamycin is a naturally occurring compound originally found in soil bacteria on Easter Island, of all places. It works by inhibiting mTOR. It's already an FDA-approved immunosuppressant used to prevent organ transplant rejection.
And the data on its longevity effects is pretty solid.
It's robust. Rapamycin extends the lifespan of mice by 10% or more, even when the treatment starts late in life. And it mitigates symptoms like decreased mobility and cognitive decline.
But the side effects related to its main clinical use, that's the major sticking point for using it as a widespread anti-aging drug.
That's the unavoidable trade-off. As a potent immunosuppressant, it compromises the immune system, making you vulnerable to infection. It can also cause undesirable metabolic side effects.
So while some people are experimenting with it off-label, the real research is elsewhere.
The vast majority of pharmaceutical research is focused on developing Rapalog-safer next-generation analogs that can capture the longevity benefits by subtly modulating the mTOR pathway without the severe immune suppression.
And finally, we have to talk about metformin. It's cheap, it's widely available, and it's surprisingly effective. How did this humble diabetes drug become a front-runner in the anti-aging race?
Metformin is the cheapest, oldest, and most widely prescribed drug for type 2 diabetes. It works primarily by activating the AMPK pathway, which is another crucial energy regulator. By influencing multiple molecular pathways linked to energy metabolism, it mimics some of the anti-aging effects of caloric restriction and exercise.
The retrospective human data here is just genuinely stunning. It flies in the face of conventional medical wisdom.
It is truly counterintuitive. Analyses of millions of patient records have shown that type 2 diabetics taking metformin often live longer than demographically matched non-diabetics.
Let's just pause on that. A group that starts with a major health problem, diabetes, outlives the healthy control group.
It's incredible. Researchers observed that the diabetics on metformin are often more obese and suffer from more comorbidities than the non-diabetics when they start treatment, yet the drug's systemic anti-aging benefits seemingly override all those disadvantages.
Which suggests its effect is powerful enough to overcome severe underlying pathology. And that's why the upcoming TAME trial is so critical, isn't it?
The TAME, Targeting Aging with Metformin trial, is perhaps the most important clinical trial in this entire space right now. It's designed to be the first major, rigorous study to directly test if a drug can delay the onset of multiple age-related chronic diseases, cancer, heart disease, stroke in a population of non-diabetic elderly individuals.
So it's not just about metformin.
No, it's about setting the regulatory precedent. If TAME succeeds, it would provide the first real evidence that aging itself can be medically treated, which could open the door for that classification change we talked about earlier. So we have this wealth of highly promising science epigenetic rejuvenation, senolytics, mTOR inhibitors, all converging on this idea that aging is treatable.
Right.
But the moment any of these cross the chasm from the lab to the marketplace, they collide with a regulatory machine that is, frankly, entirely unsuited for preventative multi-indication medicine.
Let's lay out those core regulatory impediments again, but focus on the logistical and economic reality they create.
The three barriers are all interconnected. They're self-reinforcing. First, the lack of a disease classification for aging creates financial paralysis. Second, the regulatory process is fixed on single indication approval, which creates enormous costs.
Through sequential trials.
Yes. And third, and maybe the biggest technical challenge, is the lack of validated, easily measurable biomarkers for aging.
Let's stick with that second point, the economic impact of sequential single indication approval. What kind of financial burden are we talking about for developing a geroprotector?
It's staggering. To bring a traditional drug to market already costs, on average, over $2.6 billion.
Okay, wow.
A geroprotector, which might prevent, say, 5 major diseases, would require successful trials for each one. The costs just multiply, and the time commitment extends from 7 or 10 years to potentially 20 or more years. Investors see that fragmentation and they just walk away.
So the system effectively starves itself of innovation by making the most promising category of drugs, the ones that treat the root cause, the least economically viable to develop.
You've got it. And that failure to recognize a multi-disease benefit leads us right to the unfeasible trial design problem.
Without a classification for aging and without a biomarker, how would you even satisfy the traditional FDA requirement?
Well, the traditional requirement is simple. You have to show improvement in an outcome, a definitive event like a heart attack, a cancer diagnosis, or death.
So to prove a drug-delayed aging under those rules, you'd have to give the geroprotector to tens of thousands of generally healthy middle-aged people.
And track them for decades, until they reach one of those endpoints.
Tracking tens of thousands of healthy patients for 2 decades, it sounds logistically impossible and financially ruinous.
It is. The scale and the time commitment are just astronomical. It makes the trial prohibitively expensive and largely unmanageable. This is the institutional inertia that fuels the fury we heard about in the opening.
The structure itself makes the trial of a general anti-aging drug impossible, no matter how good the lab data is.
Pretty much. So if the traditional outcome measurement is a non-starter, the only way out is through innovation in biomarkers.
Right. And how are researchers solving that technical puzzle?
Since aging is so complex, no single biomarker is going to be enough. The solution lies in developing sophisticated biomarker signatures, integrated metrics that combine multiple physiological indicators to give a clear picture of biological age.
Which is far more predictive than just your chronological age.
Much more.
Let's look at a specific example of that. Phenotypic age. What exactly is that, and what does it measure?
Phenotypic age is a really powerful metric. It integrates a small set of 9 routine clinical biomarkers you can get from a standard blood test.
So things like glucose levels, C-reactive protein.
C-reactive protein, which measures systemic inflammation, albumin, mean cell volume, white blood cell count. Researchers found that this composite score is highly predictive of an individual's remaining life expectancy and their susceptibility to age-related disease.
So if I'm chronologically 50, but my phenotypic age is 60, I'm biologically at a higher risk. The goal of a geroprotector trial would be to give the drug and see that 60-year-old biological age go down.
Or, at minimum, slow down the rate of progression. That's the key.
Yeah.
This provides a measurable, quantifiable outcome that you can achieve in a few years, not decades.
Instead of waiting for the person to die to prove the drug works.
You prove the drug works by reversing the underlying biology that leads to death. And we already have a prototype for this kind of dynamic biomarker-driven approval in another complex disease, Alzheimer's.
That's a great example. How did the FDA change its approach for Alzheimer's, and why is that a blueprint for geroprotectors?
Alzheimer's used to be treated as a static end-stage diagnosis, defined by severe cognitive and functional decline. The FDA realized that if you wait until stage 3, the damage is already done.
It's too late.
It's too late, so they modernized the approval pathway. Now they allow improvement in specific preclinical biomarkers, like a reduction in amyloid beta plaques visible on a PET scan, as a basis for approval.
So a drug can be approved based on biological evidence, even if the patient hasn't experienced full functional decline yet.
Exactly. You're intervening in stage 1 or stage 2 of the disease. It shifts the focus from treating the symptom to treating the underlying pathology.
And that approach, if applied to aging, would allow for approval based on improvements in phenotypic age.
Or other comprehensive health span metrics. It would completely bypass the need for those decades-long mortality trials.
This brings us back to the ethical core of the conflict, that concept of statistical genocide. It's a harsh term, but it reflects a deep, fundamental disagreement about risk management.
The tech and longevity community sees the traditional FDA structure as, well, morally bankrupt because it prioritizes bureaucratic process over public health opportunity.
They argue the risk of death from aging, a near certainty for billions, is much greater than the theoretical risk of a delayed drug.
Far greater. So if a geroprotector is scientifically proven safe and effective in phase 2, but the FDA demands 5 more years of tracking to meet some traditional standard,
that delay is seen as actively contributing to the death toll.
It's framed as slow-motion mass murder disguised as regulation. Every year, a bureaucrat spends debating the semantics of biomarkers is a year that tens of thousands of people die from diseases of aging that the delayed drug was designed to prevent.
It forces a critical look at how we balance the precautionary principle, that desire for safety, against the moral imperative to accelerate access.
For the most committed members of this community, the cost of regulatory caution is measured in human lives.
Given that fury and the unfeasible nature of traditional trials, it's entirely predictable that people are trying to bypass these hurdles. We're seeing 3 major accelerators operating outside or around the system. Let's start with the most visible one, the radical self-experimentation of the biohacker, exemplified by protocols like Blueprint.
This is the N1 approach. It's primarily undertaken by high net worth individuals who just reject the institutional speed of change.
Your goal is radical, quantified optimization.
Right. To achieve the health metrics of a teenager and reverse their biological age by continuously testing hundreds of biomarkers, they're essentially running themselves as private, perpetual clinical trials.
And spending a fortune to do it.
Upwards of $2 million a year to maintain the system.
What does that kind of system even look like day to day? It sounds intensely regimented.
It's absolute rigidity. The protocols involve consuming a precisely measured 1,977 calorie vegan diet, strictly managed for nutrient density, specific macros, around 40% carbs, low starch,
And over 100 supplements a day.
over 100 supplements, all precisely timed. This is combined with rigorous, structured exercise, advanced biometrics tracking, and sometimes the use of therapeutics that are often approved, but used in unapproved, highly specific combinations.
But operating in this radical N1 structure, it generates intense criticism from the wider scientific community. What are the main areas of pushback?
The first is the lack of a transparent scientific rationale. While they claim it's based on the best science, the specifics aren't peer-reviewed or published with detailed reasoning.
So critics question the choices.
They question the non-transparent rationale for very specific things, like why these particular supplements, or why are staple vegetables boiled instead of steamed when steaming is known to preserve more micronutrients. The choices can feel idiosyncratic, not globally applicable.
So it's an inaccessible black box. If it's successful, we don't know which of the 100 factors caused the success.
And we don't know if it's based on that individual's unique biology. That's the second critical point. The N11 problem.
Not statistically significant.
Not at all. Even if the person achieves remarkable results, those outcomes are highly non-transferable to the general population. Due to unique genetics, environment, what works for one person could be neutral or even dangerous for another. The sheer complexity of a 100-pill daily regimen creates an exponentially complex web of potential interactions that no one has rigorously studied.
And finally, there's the accessibility and social cost. It's not just about the millions required, it's about the erosion of a normal human life.
It creates profound social rigidity. Adherents often eliminate the spontaneous and cultural aspects of life. Sharing meals, dining out, unplanned travel.
Things that make life worth living for many people.
Critics point out that this single-minded, obsessive focus risks fostering self-involved behavior. It moves the goal away from a collective human benefit into this realm of radical personal optimization. And the intense focus on avoiding environmental impurities like microplastics risks making followers reject the outside world entirely, fostering isolation.
Okay, so if that's the private individual accelerator, the industry has its own much more scalable one, moving clinical trials offshore, often to low and middle income countries.
This trend has exploded, and it is almost entirely driven by economics. The cost of clinical trials is the biggest line item in drug development, and offshore trials offer a massive cost reduction.
How massive?
A medical center in an LMIC can charge 1/10 of the fee of a U.S. site.
Wow. And besides cost, speed is everything, right?
Absolutely. Offshore sites often allow for faster recruitment of patients, specifically naive patients, people who have less prior prescription drug exposure, which simplifies the interpretation of results. Faster enrollment means huge time and cost savings.
But the reason this is a strategic bypass hinges entirely on a regulatory loophole. It's about oversight.
That's where the systemic risk lies. The FDA accepts foreign trial data to support marketing applications in the U.S. However, due to severe financial and logistical constraints, the FDA only inspects a minuscule fraction of foreign clinical trial locations.
How minuscule are we talking?
The disparity is staggering. The FDA inspects about 1.9% of domestic sites. For foreign sites, the inspection rate is a shocking 0.07%.
And crucially, most of those inspections happen after the study is complete, not while it's ongoing. That difference creates this enormous gray zone of governmental neglect.
And that lack of oversight, combined with inadequate local regulatory infrastructure, creates a real ethical minefield. It raises profound concerns about patient exploitation, informed consent, data integrity. The infamous historical case of Pfizer testing its antibiotic, Trovin, in Nigeria during a meningitis outbreak is a chilling illustration of the perils.
Patients were enrolled, many without informed consent, unaware that free, proven antibiotics were already available nearby.
And the ethical failure was compounded when Pfizer completed the trial and then failed to provide long-term follow-up or care to the participants. They were just abandoned.
That story really highlights that offshoring trials isn't just a cost-saving measure. It illustrates the danger when globalization of research proceeds without robust oversight from the governing bodies that ultimately approve the drug.
Indeed. And it forces you to question the reliability of the data, too. Many foreign trials approved by the FDA later offered no therapeutic advantage over existing treatments or had considerable adverse effects.
And the drugs were often never even marketed to the host country.
Or they were prohibitively unaffordable, reinforcing the perception that these populations were merely used as testing grounds for Western markets.
So moving from offshore globalization to domestic modernization, the final accelerator is the use of decentralized clinical trials, or DCTs. This is essentially using technology to make the traditional process faster and more humane.
DCTs represent a fundamental transformation in the operational model. They use digital health technologies, wearables, mobile apps, remote monitoring, telemedicine to move some or all trial activities to the participant's home.
So the patient no longer has to travel repeatedly to a centralized research hospital.
Which is a clear win for participants, especially those who don't live near major medical centers. The benefits are enormous. It dramatically reduces the burden time, travel, cost, which increases patient retention and, crucially, increases the diversity of the participant pool.
And for the sponsors, the financial incentives must be huge.
Studies suggest that integrating decentralized elements into phase 2 and 3 trials could increase the return on investment sevenfold by drastically improving efficiency.
So if the benefits are this clear, why isn't every trial a DCT right now? What are the challenges?
The main challenge is global regulatory fragmentation. There's a lack of harmonization. Regulatory bodies worldwide agree on the potential, but their approaches differ significantly.
The U.S. is focused on speed.
Right. The U.S. emphasizes speed and technological integration, launching initiatives to fast-track approval for certain software as a medical device applications.
And Europe.
The European focus is often more on equity and patient education, partly driven by the stringent data protection requirements of GDPR. They're keenly focused on addressing the digital divide.
The disparity in access to technology or the knowledge to use it.
Exactly. If not managed carefully, relying solely on sophisticated digital tech could risk worsening existing health inequalities by excluding vulnerable groups like the elderly or those with limited internet access.
That's a crucial point. The promise of DCTs is greater access, but the reality could be greater exclusion if that digital divide isn't bridged.
Precisely. And regulatory bodies face huge challenges ensuring robust cybersecurity and data protection across these distributed systems. The whole field is still maturing, which slows down adoption across multinational trials. So when we synthesize everything we've covered, the incredible scientific potential of geroscience, the market-crippling inertia of the regulatory body, and that subsequent high-stakes fury from those demanding immediate access, we reveal a profound multi-front conflict.
The scientific capability to extend health span has crashed into a regulatory framework that was designed for single disease, acute problems, not for preventative, root cause medicine.
And this clash manifests in two major ways.
First, the radical, costly biohacking protocols of the tech elite, which function as private, NA1 accelerators operating outside of verifiable science.
And second, the strategic migration of industry trials offshore or the move toward decentralized formats, all designed to navigate or bypass the U.S. system's fundamental slowness.
This is a contest over time and risk.
It's between those who demand immediate access to potentially curative technology and the institutions prioritizing methodical, long-term safety. A balancing act with a devastating human cost hanging in the balance every year, a successful geroprotector is delayed.
The conflict over longevity is fundamentally a contest over control. It's control of the body's operating system, control of medical risk assessment, and ultimately, control of human destiny. The deepest question raised by this entire field is not how long we can live, or even when the breakthrough will arrive, but who gets to decide the terms of the new, updated human condition, and whether radical life extension becomes a democratized health achievement available to the many, or merely the ultimate luxury good, deepening the very global divides that this science promises to resolve.